Hey guys, in this video we're going to be talking about basics of protein synthesis and the differences between DNA and RNA. So first let's talk about proteins and what they are. Proteins are the molecules in your body that give you your traits. So for example, if you have brown hair, you have proteins that are produced by your body that give you those brown hair. If you have blonde hair, you have different proteins in your body that would give you blonde hair. So proteins are what express your traits. So the way proteins are produced by ribosomes. And in order to produce proteins, we actually have to get the code from your DNA to the ribosomes. So a couple things you'll notice here. Number one, you notice your DNA is stuck in the nucleus, and you notice that ribosomes are in the cytosol. So first of all, why is DNA stuck in the nucleus? DNA is stuck in the nucleus for protection. If DNA was found in the cytosol, the cell would actually attack it with lysosomes and would destroy it. And we know that would be really bad. So DNA stays inside the nucleus for protection. But DNA holds the code to make your proteins, which get made in the cytosol in a ribosome. So how do we get the code out of the nucleus? Well, cells use an in-between molecule called RNA. So the RNA is going to copy the code from the DNA. Then the RNA molecule can leave, join up with the ribosome, and help the ribosome create protein. All right, so what's the difference between DNA and RNA? Because they sound kind of similar, right? Well, they are similar in fashion in the sense that they both are made of nucleic acid. However, one big difference you're going to see is just in the name. DNA stands for deoxyribonucleic acid. And when you say deoxyribose, what we're talking about is the actual sugar. Okay, so here's the deoxyribose sugar that makes up DNA. In RNA, the sugar is different. It's a ribose sugar. So RNA stands for ribonucleic acid. So DNA is deoxyribose, RNA is ribose nucleic acid. Some other differences though. So here we see DNA on the left, RNA on the right. So you can clearly see that DNA is a double-stranded molecule, where RNA is a single-stranded molecule. You can see that in terms of the 4 nitrogen bases, both of them use cytosine, guanine, and adenine. However, DNA uses one called thymine, and RNA uses one called uracil. Now, some other differences that aren't pictured here. DNA is a much, much longer molecule. RNA is a much shorter molecule. DNA is stuck in the nucleus. RNA can go anywhere. And RNA is disposable, meaning if it gets destroyed, it's not going to hurt the body. Okay, so DNA, deoxyribonucleic acid. RNA is ribonucleic acid. Now, this all goes back to something we've talked about before, which is called the central dogma of biology. This is the idea that the DNA code is used to make the RNA code, which is then used to help make proteins. So again, that's called the central dogma of biology. All right, so just a little bit of background in the protein synthesis, and protein synthesis will be a separate video, but just a little background on how the whole thing actually works, okay? Your DNA molecule is used to make a molecule of RNA called mRNA, which stands for messenger. This process is called transcription. RNA then leaves the nucleus and goes and meets up in the ribosome. RNA is used to make your polypeptide, which is your protein. That's the process called translation. So protein synthesis is down to the 2 T's, transcription and translation. Always remember that translation happens later. So transcription is first, translation is later. Transcription is DNA to RNA, translation is RNA to the protein. All right, so if you have any confusion on these things, for example, the differences between DNA and RNA, you're definitely going to need to know them. So make sure you watch that part and you're able to name the differences between DNA and RNA. And just the basics behind protein synthesis. You want to rewatch this video in case you have any confusion with those. There will be a separate video that kind of goes into the deeper parts of protein synthesis.